Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome to the structural biology course. Again we are continuing with the last module, which is structure based drug discovery. We have talked about traditional drug discovery, history of drug discovery, then we come into rational drug discovery, where we are talking about pharmacophore, we are talking about accuser. We also talked about de novo drug designing, where we do the designing of a drug from scratch. Today we will put more emphasis on structure based drug discovery, which we are continuing from the last class. So, the receptor based approach is carried out in an iterative manner, proceeding via multiple computational and experimental paths until the development of an optimized lead compound happens. So, in the receptor based approach, the structure based approach, we do iteration by combining, as I was talking about, combining the structural information, high resolution structural information along with the amazing advancement in computation. So, you call it structure based drug discovery, but the best way to call it is computer assisted structure based drug discovery. The small molecule optimized in such illustrative manner, mostly having high affinity and selectivity as well as optimized pharmacokinetic properties. Now, up to now we are talking about starting from a small molecule which have a potential to be a drug to come to a position where it establishes itself as a drug. But remember, I talked about that not all small molecules with even who are able to inhibit a critical enzyme, let us say for a microorganism, is not always a good drug. To be a good drug, there are certain criteria which we will discuss in the following lectures. The developed drug candidate is then appropriate to move into phase 1 clinical trial. So, first we do the screen, let us say high throughput screening, maybe it is replaced now with virtual screening, then reduce the number, do the biochemical test, the in vitro testings, you get interesting result, go to in vivo, get good result, then come to druggability testing, the ADME tox, and if you are successful, then you are going to clinical trials. As a starting point, it is always useful to perform a meticulous analysis of structural and chemical features of the target binding site, that is the amino acid residue of the protein pocket, which means the active site, looking at the tautomerism, protonation, ionization, all sort of variation which could bring possibility to modify or optimize. Protein structures, be it in the apostate, which is ligand free, or in holostate where the ligand is bound, are experimentally determined by X-ray crystallography and nuclear magnetic resonance. We used to say that, but now cryo-EM is improving significantly and presently produce atomic level structure on a regular basis. So, yes, X-ray is historically important for long time, and even now if you want a very high resolution atomic level structure of the protein or the protein with the substrate, inhibitor and all, your best bet is X-ray. But X-ray has its own limitation of providing static structures, and today we will talk about the reason, the requirement of flexibility which will come from the dynamicity analysis, where X-ray is kind of restricted. Then comes NMR. NMR definitely allow you to go and explore the dynamicity, but you have already seen the reasons why there are limitations to use of NMR. One of the reason is when you are working with protein, as I have discussed in the NMR module, protein, especially for the big proteins, the tumbling is a problem. So, you need higher magnetic power, which means much more cost, and even if you establish a NMR facility, then also its regular consumables and all are very costly. Coming to cryo-EM, yes, cryo-EM is definitely restricted or limited in use because of several reasons we talked about, but now with more and more regular advancement, every country is investing a primary investment of 70 to 80 crore is required, but now a lot of funding agencies are coming up because they understand the requirement of cryo-EM, and if you have people who could utilize the cryo-EM facility, it would not be difficult to make a lot of innovation which is not possible with others. But there are alternatives, we have already talked about that. Protein structure using predictive modeling can be a valuable alternative, especially with the involvement of machine learning algorithm. The quality and accuracy of predicted protein structure have improved dramatically, and I have already talked about, but just to refresh your mind, in the year 2020, the last year, the machine learning based fold prediction program AlphaFold 2 have achieved a scoring which clearly says that the quality of structure prediction, the accuracy of structure prediction is at par the experimental structure. So, this is a great step, this is a great improvement. If prediction become more and more possible with, we already told there is, so there is NGS which is giving you the DNA sequence, RNA sequence. Now, if you have the machine learning based predictive modeling, you could easily get a huge number of structures that would definitely benefit the drug discovery process. Several in silico methods can be used in combination with experimental evidences to extract and organize the molecular information in order to assist the understanding of the structural and chemical basis involved in receptor ligand binding affinity and biological activity. So, now when we have opportunity to get more and more predictive structure, it would definitely helpful in developing model, be it a pharmacophore model, be it a Q-SAR model, be it what we are going to discuss today, docking based virtual screening. Receptor based pharmacophore models and molecular docking methods can be employed in early drug discovery stages for hit identification and lead generation. So, the major cutoff, the major selectivity, the major screening is required at the initial stage where you have millions of small molecule and you have to cut a good number. With the significant improvement in NGA, structural biology as well as predictive techniques, it is very possible now that these processes are coming up with more improved model. High performance computational searches are performed to screen small molecule chemical libraries that vary in size and complexity, and only a very small subset of promising compounds is selected for synthesis, acquisition and in vitro biological evaluation. So, if you remember, when I start talking about rational drug designing, one thing I was talking repeatedly that traditional drug designing was not failure, rather that we get a lot of drugs through rational drug designing. But now day by day, when we have drugs, but we need more improved drugs, the process demand a huge time as well as money. With improvement or with replacement of those high throughput experimental techniques, more and more by using computation, by combining experiments with computation, less experiment you want to do, so that you could speed up the process as well as you could cut the cost. More computer involvement, less cost. So, lead candidates can also be generated using a variety of experimental methods, including high throughput screening, which we talked about. You have a lot of, let us say, plant product, medicinal product, you bring them, you have a disease, let us say you have a specific type of cancer, you have diabetes, you have high blood pressure, you have cells and you have assays initially which we do. If you know the protein target, we do in vitro study. If you go to cell, you do cell based study and then you do mouse based study. So, that is high throughput screening. Then small scale screening of compounds that are structurally related to modulators of a target protein, structure activity relationship studies of biologically interesting molecules, and fragment based screening. Fragment based screening have changed and brought a huge success in the field of drug discovery. Let me explain this. Suppose this is the protein, the receptor, and you have the active site here. Now, a drug have to bind here is potential small molecule and it have to bind here. So, it is very difficult to find a drug with A, B both the moiety and C and D connectors. Instead, if we target A and B separately and then link out them, then the chance of drug discovery become hugely enhanced. So, that is where fragment based drug discovery is coming out. Focused libraries can be designed based on new bioactive molecules through the incorporation of new data sets of compounds sharing a high degree of chemical similarity as well as interesting structural diversity. The molecular and biological properties of new compounds which are related to structure can be further rationalized and even predicted in terms of their chemical modification as a consequence of their direct interaction with the target receptors. Biochemical, crystallographic and spectroscopic methods are useful in determining the binding properties and mechanism of action of promising ligands. So, once you get the hit, you do all those biochemical, biophysical studies, crystallography is end, but by using low resolution techniques which we talk about, let us say circular dichroism, let us say UV-Vis, let us say UV-Vis based kinetics, let us say binding processes like ITC, isothermal calorimetry, SPR, surface plasmon resonance, you could do a lot of screening and get lot of idea about the receptor ligand interaction. The knowledge which is generated in point of view of chemical based knowledge and biology based knowledge from this step is a key component in medicinal chemistry and can be used in the iterative design of new ligands with improved properties characteristics. So, what it is talking about, before we get some success, but we do not have this biology to chemistry to structure all the interdisciplinary connections. So, you get an activity and you are happy that this is a good drug. Now, you get the initial success, but you are not happy with that, you try to get the structure, interaction, chemistry and then make a computational model, do more iterative searching, do more iterative modification, so that the modified ligand would be more specific, more optimized towards the target. For this purpose, 3D quantitative structure activity relationships, 3D QSARs, are among the most important strategies that can be applied for the successful optimization of leads. In this context, 3D QSAR models are generated to explain the relationship between the intermolecular interaction related to the 3D conformation of a set of structurally related molecules and their experimental activity. For example, IC50, Ki, which is inhibitor constant, therefore, providing a rational basis for the development of new promising compounds. One more thing I want to talk about. So, after a few slides I will talk about that. Structure based approaches have increasingly demonstrated their value in drug designing. The impact of these technologies on early discovery and lead optimization is very significant. Although there is a multiplicity of different approaches being employed in early stages of drug discovery, structure based drug discovery or SBDD is one of the most powerful techniques and has been used quite frequently by scientists in the pharmaceutical industry as well as in academic laboratories over the past 50 years. Structure based drug discovery approaches continue to drive important advances in drug design, integrating traditional and modern technologies from the field of medicinal chemistry, computational chemistry, informatics, biology, biochemistry and structural biology. Structure based methods bring the 3D structure of protein to light and thereby greatly enable many drug discovery effort to identify novel small molecule drug candidates that selectively and potently modulate the right biological target. The ability to make knowledge based decisions during the early phases of drug discovery is the key to decreasing hit to lead and lead optimization cycle times. So, what I am trying to say is, see, we are talking about traditional drug discovery, we are talking about rational drug discovery. In the traditional drug discovery, there are many different processes. In rational drug discovery, we say ligand based drug discovery, structure based drug discovery. In the ligand based drug discovery, there is pharmacophore modeling, QSAR, but if you remember, when I was talking about molecular modeling of protein and I talked about the package ITASR, which is actually claimed to perform predictive modeling using ab initio approach. But if you remember, you go back to the module and you see that a lot of different approaches are integrated here. Similarly, now for better drug designing, we are not restricted to a certain bit of process, we are merging all of them and using what is needed on for that step. So, that is one, like a combinatorial approach is growing, a interdisciplinary approach is growing more and more. The evolution of medicinal chemistry has resulted in an increase in the number of successful application of structure based approaches. The importance of these approaches in exploring the chemical space of biologically active compounds is well established, considering that these powerful strategies have significantly contributed to the discovery and introduction of several new chemical entities. NCEs are new chemical entities into clinical trials for a wide variety of therapeutic applications. But more importantly, it helped drug designers to reduce the time as well as the cost of the process. So, we are talking about different things, but in one word, if we want to understand that, it is not very different. Traditionally, we collect in the Chinese traditional medicine, in Ayurveda, we look at animals, we look at birds, because people who are culturing those used to be living in the forest. So, have close relation between like them with animals. So, they look at, they study the behavior of other organisms and see how they are finding a way out for surviving from a disease, and then they collect that, make them better and continue. So, that was traditional. Now, when this traditional culture wants to be industrialized, the industrialists, what they did, they innovate high throughput instrumentation, they involve a lot of people, and then they start doing screening so that they could get the desired leads. But now initially there are some easy hits, we say low hanging fruits. So, that was obtained already, and now the scenario was becoming more and more difficult. It like the easy targets are already achieved. So, now what we have to find out, what we have to explore is difficult targets that takes much more time, much more manpower as well as much more cost. So, now we are gradually replacing those high throughput instrumentation, involvement of more and more people, involvement of more and more experiment using the computer. Be it come from the molecular modeling, be it come from the docking, which we are going to discuss more in this class, be it the machine learning based involvement, training set development, all are actually replacing the experiment with theory. So, in structure based drug discovery, you have target structure, and when you get the target structure at high resolution, you would be able to characterize the binding cavity, let us say the active site of an enzyme, that will help you to like design interaction, go for heat identification, and ultimately towards lead discovery. Now, once you discover a lead, you are not being limited there, you might go from there to directly to biological assays, market, but a much better way is to go from lead discovery to lead optimization and ultimately get the drug candidates going for clinical trials. So, high throughput screening, as I told, now converted to focus screening, which means we have to perform less synthesis and less biological assays which are costly, as well as the biochemical assays. We do receptor based virtual screening which generate 3D pharmacophore models. So, you see the virtual screening is connected to pharmacophore model, and then structure activity studies, docking, high resolution crystallography with receptor based 3D QSA studies. So, in that way we are in this side there are experiments costly, we are replacing them with computation. But we have to also remember, if you remember in the last class I was talking about, if you make a compound which kills a rat, a mice, and you think that this is a drug, you cannot say this is a drug. Drug is something which you should consume. It should not have side effects, or at least it should not have a lot of side effects. A lot of drug have some side effects, but it should not kill you, right. So, that is a challenge. You do everything and then you get a drug, you are happy because you could make money out of that, but then you do admetox and it says no, it is a poison. So, the entire money your company have invested is now go to when? And that is why there is something people start about thinking, is it possible that we could develop something, the admetox what we are performing experimentally, can we do something so that we could do at least a set of screen at the initial level, so that a small molecule we could at least say that it is a druggable molecule. That druggability is answered by in silico admetox, save a lots of money. We will discuss about in silico admetox in our next class. So, methods for rational drug discovery, the structure based drug discovery, de novo drug designing which we have already discussed, and now docking based virtual screening. Here our focus is docking based virtual screening, all the factors influence them, and then in silico admetox. So, today we will discuss about docking based virtual screening. Next class we are going to discuss in silico admetox. What is virtual screening? Virtual screening is a computational technique used in drug discovery to search libraries of a small molecule in order to identify those structures which are most likely to bind to a drug target, typically a protein receptor or enzymes. Definition never like help people understand. So, let me explain. Remember what I was talking about, in traditional drug discovery there was something called high throughput screening. Now, high throughput screening is expensive, it need instruments and it needs manpower. So, people start understanding the process of high throughput screening and try to replace it with computational process, and that is called virtual. So, you do the screening in a virtual world, that is why it is called virtual screening. Virtual screening has been defined as the automatically evaluating very large libraries of compound using computer programs. As this definition suggests, virtual screening has largely been a numbers game focusing on how the enormous chemical space of over 10 to the power 60 conceivable compounds can be filtered to a manageable number that can be synthesized, purchased and tested. So, let us say you want to work in a drug designing project. If I give you, let us say, not even very large, 1 lakh compounds, and you have to synthesize all these 1 lakh compound. It is actually very difficult, it will take years for you, and that is where virtual screening is coming. It helps you to screen initially, making a computational model, and then when the number cut up to a achievable one, then you synthesize and do further in vitro, ex vivo, in vivo testings. Although searching the entire chemical universe by itself is an interesting problem. So, when I talk about these, even achieving this computationally is a very interesting phenomena. More practical virtual screening scenario focus on designing and optimizing targeted combinatorial libraries and enriching libraries of available compounds from in-house compound repositories or vendor offerings. So, what it talks about? So, you know a structure, you know the proteins, you know their charges. Based on them you start design. So, you get a designed lead and then you make many derivatives, then screen them and then you see that available library is there or not. If not, you synthesize them in house or ask if some pure derivatives are available or not for testing. As the accuracy of the method has increased, the virtual screening with the information coming, with more and more compounds coming in the database, with more and more structure is solved, with more and more biochemical, biophysical informations are there, with more and more predictive modeling happen, the accuracy of virtual screening increased. It become an integral part of the drug discovery process, because it is what we dreamed about. Virtual screening can be used to select in-house database compounds for screening, choose compounds that can be purchased externally and to choose which compounds should be synthesized next. So, now we will take a look about virtual screening and how we could utilize it in structure based drug discovery. So, as I told, in structure based drug discovery there is a target structure, but as I told we will mix. So, virtual screening is possible with unavailability of structure, where the structure is unavailable, like alignment, which is superposition field based similarity, that could be a process. Descriptor based, we have already talked about descriptors while we are talking about Q-zer. Graph based, we talk about the graph theory. We have a binding site, we get the points and then we connect them, we get the pattern, and that is where graph theory works. Pharmacophore, we have already talked about a lot, and shape based. Whereas known structure, how structure could be known, we all know now. There is X-ray, there is NMR, there is cryo-EM, but also there are many biochemical, biophysical, kinetic methods which include information how actually the interaction between the receptor and the ligand is happening. But now we know that the prediction of model structure is also getting more and more accuracy, contributing hugely to the known structure based analysis. And here it is docking based virtual screening, what we are going to talk about. So, what is docking? If you remember, I am sorry I did not put the picture would help you remembering. When we were kid, we had a setup of toy where you have a base and you have different shapes, and what we used to do, we try to get the exact shape to the base where there is place. So, these help us understanding the different shapes there, how they different, how they match and all. To me, in a simplistic way, docking is matching. It is true that there are a lot of improvement going on which we are going to discuss, but at a initial conceptualization you could think about the base is the protein and you have shapes and there are places where you could match the shape. So, finding a proper match is a process of docking at the initial level. Also, if you try to understand, you will get that one of the major base to understand drug designing is the interaction between protein and ligand. So, docking is initiated to mimic that, and that is why when you know the structure, this is the method of choice. So, docking attempts to find the best matching between two molecules. It includes finding the right key for the lock. Given two biological molecules, determine whether the two molecules interact. If they interact, what is the orientation that maximizes the interaction while minimizing the total energy of the complex. I repeat, is it interact or not? If interact, what is the orientation that maximizes the interaction while minimizing the total energy of the complex? The goal of docking is to be able to search a database of molecular structure and retrieve all molecules that can interact with the query structure. So, this is the flow chart to give you a simplistic idea of docking. This is not a bioinformatics course, so that I could go in details, but I could talk about so that you could do that. So, you have the target protein which would be present in .pdb format, and I am sure now you understand what is .pdb. The ligand should be in .mol2 or .pdb format. Okay. First, you have to add hydrogen and charge. When you do that, when you add hydrogen and charge, both of them, protein and ligand, would come to .pdbqt. This is very important because docking works only when they are in .pdbqt format. .pdbqt is docking specific format. Now you could optimize bond, angle, especially if you remember in case of ligand I talked about rotable bond, I will talk about that also, because if you think a molecule have more rotable bonds, so it could change conformation, so it comes flexibility issues and all, we will talk about them. Then so you have them, then you set the parameter, the grid formation, define the receptor coordinate, grid spacing, grid center, exhaustiveness is the redundancy and number of output poses. Then you run the docking, you get the docking result. This is not the end. You have to select the best pose for protein ligand complex compared with experimental data. So, how you do that? How to choose the best complex is one of the biggest challenge of docking. So, continuing, in the field of molecular modeling, docking is a method which predicts the preferred orientation of one molecule to a second when bound to each other to form a stable complex. So, when you want to do drug designing, you do docking. So, this is the process of docking, and then you have to choose by getting the pose and score it. So, the basic requirements in docking is 3D structure of the protein, library of a small molecule, because you have to run them, and selection of proper protein ligand complex. This you understand, this we understand, we need a library I talk about, and more importantly this selection of proper protein ligand complex through scoring and posing. One thing left. So, docking or molecular docking is the unit operation. What I mean by, you have a receptor, you have a small molecule and you get a complex. Now, if you want to repeat it, you put a program so that it run in loop. So, using molecular docking as a unit operation, if you want to repeat the same operation instead of one molecule with a library, this is called docking based virtual screening. Is it clear? So, what is docking? In docking, you have a 3D structure of a protein, you have a 3D structure of a small molecule, you set them, you make PDBQT, then you run docking, and you have criteria to get the best pose and best score. Now, if you repeat it, if you put it in a loop by taking a library instead of a small molecule, then that is called docking based virtual screening. Major challenge is to rank the molecule to select and proceed further. This require a good scoring function. So, I have given a lot of docking softwares. AutoDock is the software which most of the people use, but you could use AutoDock, you could use Maestro. Maestro is a commercial one. Gold, AutoDock Vina, again it is from AutoDock, and AutoDock Vina is from Scripps. They are freely available. FlexX, Dock, Hex. There are other ones which are web based docking servers. I have given the links: ClassPro, GramX, JetDock, PatchDock. You go there, you provide your protein, you provide your small molecule, they will automatically do the docking and providing you. So, from our discussion, I could say that docking based virtual screening, few things are very important. One, the library of small molecules, how the small molecules should be, from where we will get them, how we could separate them, what type of molecules we could pick up, how we could relate our lead with the library, all this you get answer in getting a library. As I told, posing and scoring is very important, but then most of our consideration of docking, again if you remember the kids matching, is based on rigid docking. With advancement in science, with the advancement in computation, we want to explore the flexible area. So, flexibility in docking is a very interesting area, and when we talk about flexibility, we cannot talk without involving the molecular dynamic simulation. So, finding lead compound. A lead compound, as I have talked earlier, is a small molecule that serves as the starting point for an optimization involving many small molecules that are closely related in structure to the lead compound. Many organizations maintain database of chemical compounds. I have talked about zinc database, ACS database, some are commercial, some are public, like freely accessible. Now, when you are thinking seriously about molecules, let me tell you, the whole world of molecules are divided into majorly two parts. One is called real molecule. How the real molecule comes? You see around you there are lot of synthetic labs, there are companies, there are drug and pharmaceutical industries, they keep synthesizing compounds. A lot of synthetic lab, they optimize synthetic protocols, they work on new synthetic process. So, whatever they synthesize, they publish it, and all these compounds are actually exist in the world, they are called real molecule. So, 1 to 2 millions in-house archives of large pharma and agrochemical companies, if they isolate, if they synthesize anything, they are real compounds. 3 to 4 millions of samples available commercially, so all of them are real molecule. Hypothetical molecules are, let us say you get a lead compound, then you do all type of modifications and design a lot of molecules library. This library does not exist, but at least it have existence theoretically. They are called hypothetical molecules. I hope it would be clear. So, coming to small molecule database library, small molecule libraries can be divided in majorly three groups, which are general, focused and targeted, but there is a special one which is cherry picked or exclusive library. So, general libraries are, as the name says, general, designed to be a broad interest for high throughput screening against any target. Suppose a pharma industry have identified a lot of medicinally important plants and they isolate the small molecules. So, they do not know what exactly they are destined for, but they think that it could be tested with many diseases, they are called general library. Focused library, the focused libraries are aimed at a family of related targets, they are not specific for one protein, they are targeting a family. For example, a library designed against chymotrypsin fold. You know that chymotrypsin is a protease. So, there is trypsin, there is chymotrypsin, there is elastin, there are a lot of such cetylizin, there are a lot of such proteases, and because all those proteases work through serine as a catalytic residue. So, if you could inhibit them with a library of compounds, they are called serine protease inhibitors. And then come to targeted libraries which are of interest for a drug designer like me or a pharma industry. They are aimed at a single therapeutic target such as HIV-1 protease like beta-lactamase. Cherry picked library is an exclusive library of a set of compounds. So, they might be destined, they might not be destined. You ask a pharmaceutical company to synthesize them for you, and you signed a contract that they could not sell those library to other people. This is called cherry picked or exclusive library. These are database of different small molecule libraries like PubChem, ChemBell, Binding DB, Zinc, ChemSpider, Drug Bank, GRAC, GRAC, Cambridge, Maybridge, ChemDiv, Live Chemicals, Fakes, Enamine. So, you could go, you could look at the links. I have made the links available to you. More interested you are, go through them and you could find all those available libraries. If you see the word written public, they are freely accessible. The ones which are commercial, you have to pay for them. Coming to scoring function. So, scoring function is an approximate mathematical methods used to predict the strength of the non-covalent interaction between two molecules after they have interact. So, if you see it is an energy profile, you analyze those and then you get the base docking mode. Before going into details, scoring is a very challenging job. Why? You guys understand within a minute. If I ask you, how many of you thinks the scoring of examination which you are going through is a correct evaluation of each and every individual and their hard work? Many of us do not agree to that, but because the administration, the government make it a rule, so you have to go through that, you have no choice. If you would have choice, a lot of us could have explored different possibilities, right. Here we have choice because in exam we get some quantitative number, we get some numbers and compare. Here to get a success, of the drug designing, we have to make it optimized, and the optimization needs comparison, comparison needs scoring. That is why perfect scoring is a very critical and challenging thing. So, what is scoring? For all molecular docking programs, one important problem is to develop an energy scoring function to describe and estimate protein ligand fitness rapidly and accurately. Better scoring function you have, more straightforward scoring function you have, quicker the drug designing. More accurate scoring function you have, better your success in drug designing. You understand? Scoring functions are mostly used for post docking analysis. Dozens of scoring functions have been published since the early 1990s. Theoretically, a scoring function is expected to represent the binding affinity between ligand and target macromolecule by calculating the non-covalent interactions, the hydrogen bonds, the salt bridge, the hydrophobic interaction, base stacking, metal binding, anything, and providing basic character like speed and veracity. Scoring functions are generally classified into three basic types: force field analysis, empirical scoring function and knowledge based scoring function. Force field analysis, if you remember your 10th module of MD simulation, I have shown how a equation of a force field could be developed taking the necessary factors, like if you go back and check, we have taken bonds. For bond we take stretching, for angle we take bending, for torsion we take dihedral. These are covalent. For non-covalent we take hydrogen bond, van der Waals and Coulombic. So, taking this 6 factor, we develop a equation and apply classic physics to calculate the movement. So, that you could bring here and calculate the energy. Then you could also develop empirical scoring or statistical scoring functions. Then you could also design knowledge based scoring function, like suppose you have a protein and you have its known substrate ABC. So, you take ABC, you know the structure, you do docking and you use RAP method like statistics or something to calculate the energy. A is let us say 5, B is 7, C is 10. Then because you know both of them, you do experimental binding affinity. Now you get A is 10, B is 14 and C is 20. So, you correlate them and you know whatever you get here, this could be double. So, in that way for unknown ligands you design. So, you get a knowledge here and convert them to scoring the unknowns. Getting the knowns, do both experiment and theory, correlate it, use those data to plot a graph and now use the unknowns towards getting the real value. Force field analysis can calculate the actual molecular forces that exist in the protein ligand complex by optimizing, as I already talked about, van der Waals, electrostatic and hydrophobicity to each force field. Gold score, CHRM, AMBER, OPLS are such function I have talked about. If you go back in the MD simulation, you will see those already. However, because it is an extremely complicated progress to predict the binding free energy of protein ligand complex, if you remember I talked about how complicated and computer intensive they are. Only limited success have been achieved so far in this approach. On the other hand, researchers attempt to apply the other progresses such as modern force fields, quantum mechanics methods, solvation models into the predictions to achieve technical breakthroughs. Empirical scoring function is the fastest one among all of the scoring functions. According to the developing algorithms, empirical scoring function optimizes empirical terms such as van der Waals interaction energies, hydrogen bonding energy, electrostatic energy, hydrophobicity, dissolution, interacting atoms of binding molecule and change in solvent accessible surface area or SASA. TS Chem score is a new empirical scoring function which is validated on 14 protein targets. It improves the correlation between the calculated scores and experimental binding affinities compared with the other scoring function. 30 compounds which are scored by TS Chem score as JAK3 and YOPH inhibitors for experimental validation. 6 active compounds were selected, confirmed as JAK3 inhibitors, and 4 were confirmed as YOPH inhibitors. So, empirical ones are rough, they are quick, but generally their values does not make much sense, whereas as you see in the chem score, the modern developed empirical scoring function is actually working, which is a great sign. From the results we can find that TS Chem score shows a better correlation in virtual screening in comparison to the existing scoring functions which we have worked up to now. Different from the first two group of scoring function, knowledge based scoring function model behavior of protein ligand uses experimentally determined structure geometrical data from databases like PDB, as I have explained earlier. This kind of scoring function converts structural information from protein ligand complexes into distance dependent pairwise potential. A good number of studies have compared different docking programs and their score functions to evaluate their accuracy in correctly scoring and ranking ligands that bind to a protein target. Current scoring functions have been greatly developed, however, none of them is perfect in terms of accuracy in hit ranking and general applicability. This is a huge research font. If we could make it perfect, it would be possible to invent, it would be possible to discover a lot of drugs in very short time. Every scoring function has its advantages as well as limitations. Combining the results of multiple scoring functions and forming a consensus has proved to be a beneficial method in scoring and ranking compounds to improve the probability of finding correct solutions. This is a very important thing. I always tell that to my students, and I am telling you, whenever you are doing computational in silico methods, never be restricted yourself to one procedure, because as I have told you, from 1990 a lot of people are working and a lot of people are coming up with different force fields, modified force field, different process and all these things. And when you are going to be a user, in most of the cases you have no idea what are the physics used behind. So, what is my suggestion? My suggestion is, if you are taking help of those softwares for initial studies, take 4-5 such functions, apply and see the correlations. If you see that there is a data consistency, then proceed. That would be extremely beneficial. Consensus scoring supposes that each scoring function may not be highly accurate, but has at least some predictive value for compound ranking on a specific target. A variety of statistical methods such as mean, median, minimum or maximum of ranks of scores are used to generate the values from different scoring functions. Those are extremely helpful to discriminate the more accurate binding affinity of ligand receptor complexes. Tools for consensus scoring functions include multi-score, X-score, GF-score, select CS and consensus doc. So, going for improved description of binding energetics coming with a polarization effect. The continuous and vigorous development of additive force fields for protein and small molecules plays a fundamental role in the correct evaluation of relative binding affinities upon docking or MD base binding or unbinding protocols. So, as you now understand, to get success in the field of drug discovery, scoring is the most critical factor. So, a lot of research is going on here. Already there are developments. Furthermore, efficient tools for automatic atom typing and parameterization of large libraries of compounds have been developed. So, this is very interesting. Suppose this is your lead compound, your lead compound is phenol. Okay. Now, with research you know that a positive group is good here, a negative group is good here, a hydrophobic is good here, and blah blah. So, if you have a simple computer program which will allow you to introduce options here and then make all possible permutation and combination from one small compound, you get around 1 million compound containing library where the lead compound is this. So, this is automatization. Also with that the parameter of the molecule, if it could also be generated automatically, they would be extremely beneficial. Despite the robustness and accuracy of classical force fields, a number of energetic effects of binding remain challenging to be described by energy functions based on fixed partial charges. Key backbone and or side chain groups can be treated as quantum mechanical probes to accurately approximate the local electronic structure in the binding site. So, one of the reason when we are doing MD, we are doing classical physics, you all know, but when we are considering the small molecules, here we do not need to go for classical physics, Newtonian physics, we could go to quantum mechanics where the calculations would be pretty accurate, you do not need to do the experiment. Briefly, quantum mechanics probes use semi-empirical Hamiltonian for the evaluation of interaction energy between ligand and individual polar group of the protein to reproduce polarization and charge transfer effects. One advantage is that partial charges are not needed. The main drawback is that dispersion effect are not captured by efficient semi-empirical method. You have to go to more classic quantum approach. Alternatively, application of polarizable force fields is expected to improve the description of binding energetics in the coming years. What is polarization force field? So, once you are talking about, I am giving very simple example, OH, okay, phenol. Now, when it could be, when it become O minus, it could resonate, right, it could develop a quinone type structure. So, those change dynamicities polarization effect. So, as I told already, post selection where most of the current research is focused, more sophisticated scoring functions takes longer in terms of computation, balance need for speed versus need of accuracy, and virtual screening needs to be very fast, because with our ability to make it faster, more number of compounds would be included in the screen, and more number means more chance to get success. Now, we will move towards flexibility. If you look at the docking scenario, historically it was rigid receptor and rigid ligand. From there it shift to rigid receptor, the protein is still rigid, and flexible ligand. If you look at modern day docking programs, as I have listed, but if you have to ask me to name, if you go to Autodock, you will see that you could fix or allow the rotable bonds to rotate or not to rotate. So, you could make the small molecule flexible, but you cannot make the protein flexible because it is big in size, your calculation would be much more intense. Then you might have flexible receptors and rigid ligands. This is not a very common biological scenario, but if you consider chemical modifications in chemical engineering, nanotechnology, you find this scenario where the receptor is flexible and the ligand is rigid. And now what is our dream? We want to go there, flexible receptor and flexible ligands. But before ending this up, so this rigid receptor rigid ligand is historical, where it started, where we had lock and key concept. But this is not far away. Now, if you look at Autodock, you could change the movement or rotamer conformation of side chains of amino acids which are involved in docking. So, that gives certain amount. So, what I mean, you have the receptor, you could make these flexible. So, that is kind of our journey towards. So, protein flexibility constitutes another challenge of structure computer aided drug discovery. Since the advent of structural biology methods and their application in structure based drug discovery, it has become apparent that many protein binding sites cannot be represented as a single snapshots. Once we started our journey as a drug designing historically, our base was the high resolution X-ray crystallography structures, because we start getting X-ray crystallography structures since 1960s. So, we thought that this is the representation of protein, it is a static representation, but with more advancement we start understanding that no, protein moves, protein breathes, and those small change in conformations are extremely important in drug designing. Significant structural rearrangement takes place to accommodate diverse ligands. The pioneering work of Wells and co-workers targeting the interleukin 8 established protein-protein interactions as challenging targets to comprise flat and featureless surfaces that are inherently flexible and adapt to different binders. In this context, I would also introduce something called IDP. You guys now, if you search Google, you will see internally disordered proteins. There are proteins, especially very important proteins, which use kind of as a platform in very critical biological event, let us say replication, transcription, or in this type of process we talk about 1 or 2 or 3 proteins, but actually hundreds of protein comes and takes part, and how they switch, how they specifically interact is because there are internally disordered proteins who are like proteins, as I talked about in the entire course, is getting into the energetically stable state. So, generally protein gets a stable fold, not with the internally disordered proteins. Internally disordered proteins, they take shape only when they interact with a specific partner. In that way, they could interact with A in one way, they could interact with B in other way. So, as I told, in a large biological macromolecular assembly where switching in, switching on are happening continuously, a set of protein is coming, a set of protein is leaving, all those are majorly controlled by internally disordered proteins, and those are real challenges in terms of drug designing. So, since the discovery, several other examples of protein flexibility upon ligand binding have been shown. Cryptic pockets could be discovered by accident or through detailed analysis, often by more than one independent approach, as in the case of the polo box domain of polo-like kinase 1, a mediator of phosphorylation dependent protein protein interaction and cell cycle dependent cancer target. It has also been shown that structural adaptations are not limited to protein protein interfaces. The enzymes are also more plastic than originally predicted. As I was talking about, the enzyme, we develop drugs based on its substrates and products. But then we come to understand the transition state theory. An enzyme is not destined to perfectly bind its substrate or product, rather its intrinsic property is to stabilize the transition state. I will talk about that in the case study part. So, enzyme substrate binding have two models. One is the lock and key model, which was initially taken, but now it is induced fit, where substrate or ligand, when it bind to the enzyme, the shape changes. Also, there is concept of cofactors, one receptor, one protein and several ligands. The enzyme may bind several ligands in different places. If the protein binds to several ligands and the affinity for binding ligands 2-3 changes after the first ligand is bound, the binding is said to be cooperative. If you look at metabolic pathway, by metabolic pathway I will give you the very known and common example of glycolysis. You see there feedback inhibition. So, the cooperative interaction might be good, like we see in case of hemoglobin, as the monomer binds, then come the dimer, more oligomerization, it is more speeding up. So, it is positive, but in feedback inhibition it is negative. So, positive cooperativity, the affinity increases with ligand binding, negative cooperativity, the affinity decreases with ligand binding, and if there is no change, the binding is said to be non-cooperative. So, as I told, the cofactor may induce the fit, and that is called allosteric. As you could see, when the cofactor bind, the binding induce conformational change and then the ligand bind, this is called allosteric. So, predicting binding computationally, as I told, lock and key is the simplest case to predict. Little or no flexibility need to be modeled, 6 degrees of freedom, 3 rotation, 3 translation. Induced fit is much more difficult, much greater than 6 degrees of freedom. Algorithms may need to model the movements of the side chain and backbone of the receptor and ligands. Now, here you see you are considering your protein and then a small molecule is coming. So, you get the degrees of freedoms. Think about if both are macromolecule. So, both have number of possibilities. So, the possibility they could interact with each other is a absurd number, and that is why it is so difficult to study protein theoretically. And remember, I told about the simplest idea of docking, that is why I told about, because docking cannot validate those complex situations. So, information on binding site flexibility available from experimental or simulation studies can be taken into account to prepare one or more conformation of the target protein. So, yes, you cannot model all possible conformations, but you try to get more structures, and if you get multiple conformation, you include both of them in the docking ensemble, and in that case molecular dynamics helps getting receptor flexibility. So, MD simulations have been long proposed to provide insight into protein dynamics beyond that available crystallographically and unravel novel cryptic binding sites, expanding the druggability of the targets. So, I have talked about already when we discuss MD simulation, you include MD simulation, though you remember that MD simulation is mathematically very intense, but introducing this would help you looking at the dynamicity, and we all know even a low resolution movie is much better or much truthful than a high resolution picture. One of the first described approaches was the relaxed complex scheme, which combines all atom nanosecond long MD simulation of the protein target to describe its conformational flexibility with rapid docking of small molecules to the protein snapshots saved along the MD run. So, you do a classic MD run, you get the ensemble, you look at the flexible conformational flexibility, and then you take different conformers and do docking. This scheme has been a number of successful application, including one to the cancer relevant MDM to P53 interaction of HIV integrase, or so one is MDM2 P53 interaction, another is HIV integrase. Rewardingly, the cryptic pocket first proposed in silico in the latter case has been then validated experimentally and is exploited by enzyme inhibitors approved for use against HIV infection in the clinic. While the idea is conceptually correct, often the conformational change needed to uncover the pocket would not be observed at the 1 millisecond time scale accessible to explicit solvent MD simulations with conventional protocols on commodity compute clusters, highlighting the need for more sophisticated sampling schemes. So, I could explain this in a easy way. You know that protein have a lot of local minimas, right. So, in most of the cases it traps there and just you do not see the changes. You cannot push it from one minima to the other or local minimas to the global. Is it possible? If you remember, it is possible with biased simulations, like one I have discussed is simulated annealing, but there are many other methods like replica exchange method, like metadynamics. So, there are bias methods also. The molecular dynamics induced feed MDIF protocol makes use of atomistic simulation to increase or reduce the aperture of sub pocket in the active site. Another approach to the long time scale problem predicted on the fact that cryptic sites are more likely to open in the presence of their cognate ligand, and several groups have proposed to perform ligand mapping simulation. So, you have the protein, you see the dynamics of the protein, the flexibility of the protein, then add the ligand and see where the flexibility is reduced. This includes the MD based protocol called SILCS, site identification by ligand competitive saturation, which uses high concentration of the small molecule ligands to map possible binding sites on protein surface. MD simulation of ligand binding and unbinding. The application of MD to drug discovery projects is expanding following the need for insights into binding, unbinding and conformational change events at spatial and temporal resolution that is not available experimentally. MD simulations can be used to map ligand binding sites and analyze unbinding pathways. Extensive MD simulations have been performed to determine binding sites and bound conformation of allosteric inhibitors of the M2 muscarinic acetylcholine receptor. These simulations have revealed a new binding site dependent on cation pi interactions, which is placed 15 angstrom away from the classic recognition site and was validated by radioligand binding experiments. Unbiased simulation of ligand unbinding are useful not only because they provide insight into affinity of the complex. So, you have already known information is good, but then if you want to see a blind way of looking at the interaction of protein and ligand. It is also possible to study the dynamics of complex formation and dissociation and quantify complete energy landscape and kinetics for the processes. It has been reported that the unbinding rates are strongly dependent on the state of the protein receptor, which in turn depends on the condition of the experiment and in particular the concentration of the ligand. Therefore, as the unbinding of small ligands can be orders of magnitude faster than relaxation of the protein, it is crucial to select the right conformational state when executing high throughput docking campaigns. MD simulation for ligand optimization. MD simulations are used frequently to guide further optimization of the molecules stemming from in silico discovery campaigns, particularly in the absence of a crystal structure of the complex with the target protein. Even if the structure has been solved, MD may provide insight as to which interactions are stable over time and contribute mostly to binding. As I told, you have the ligand, so you have the protein structure high resolution, you have the receptor, you have the lead. Now, you make lead derivatives and do simulation, see the interactions as well as binding energy calculation. This way, in a campaign to develop compounds targeting the bromo domain of CREBBP, MD simulation revealed the amide linker of an initial hit compound as not contributing directly to binding and thus replaceable. In other example targeting EPH before tyrosine kinase, MD simulation were used to prioritize the docking hits that were maintaining a stable hydrogen bond network in the protein active site, which were then validated as true inhibitors with nanomolar affinity. So, there is a correlation coming up, there is a protocol developed now going through. So, coming to the summary of the class, structure based drug designing or drug discovery is going through a critical juncture, a very prime time where advancement in NGS, next generation sequencing, machine learning in biology and significant enhancing in the computational power help expedite the discoveries as well as exploring in higher space, higher numbers. Two main techniques seem to emerge in the contemporary application of protein structure based computer assisted drug discovery methods. They are particularly useful in the initial phase, which is ligand identification, and also advanced phase ligand optimization of drug discovery projects. For ligand identification, high throughput docking of large libraries of small molecules up to 10 to the power 6 molecule of 10 to 25 non-hydrogen atoms each to a rigid protein structure is a method of choice. Importantly, classical force fields with implicit solvation and endpoint calculations, that is the evaluation of binding energy using only one structure of complex, have shown substantial predictive power. So, you need 4 structures, 5 structures, but when you are doing simulation, you are actually going through the ensembles, you are actually going through the structure space. For ligand optimization, MD simulation based free energy calculations can be carried out on small set up to few hundred of related molecules. So, once you make the initial cut, you do further studies coming into 100 and do that process. The prediction of relative binding affinity by explicit solvent MD simulation is more accurate the higher the pairwise compound similarity as statistical convergence, that is sufficient sampling is easier to achieve if the binding mode does not change substantially. If it is very flexible, if after binding even it is not stable, then it is a problem, otherwise it is totally good. The MD based protocols include free energy perturbation, FEP, and thermodynamic integration, TI, alchemical free energy calculation, umbrella sampling for the theory and for successful application. Furthermore, MD simulation of spontaneous unbinding or binding can provide atomistic information on pathways and kinetics and have shown potential for the identification of allosteric sites. So, if you remember, I was constantly comparing with the high throughput, the traditional method. In the traditional method, if we get effects, we do not know what is happening, but now with high resolution structure we could see, but also in the MD simulation each and every change, their effects, their interactions, their energies, everything is in front of you. So, it is much easier for us to take accurate decision. While the rigid protein docking of large libraries of fragments can be carried out on a conventional desktop or even laptop computer, enhanced sampling protocols and the availability of a compute cluster are essential for the MD based calculation of relative free energy and binding kinetics. The continuous development of graphical processing units, the GPUs, and establishment of more supercomputing centers in regular basis also benefiting the MD based protocol substantially, because now we have access to higher computational power. So, with that I would make an end of this description of structure based drug designing, mainly docking based virtual screening. So, though I talked about virtual screening, I talked about docking based virtual screening, eventually we have to understand that everything is mixed. So, we take help of pharmacophore, we take the knowledge of QSIR, we use MD simulation, everything coming together giving us the opportunity to go for better drug designing. In the next class we will talk about how our knowledge of ADMETOX is modeled and computationally we are able to screen the drugability of the drug molecule, the small molecule. Thank you very much.